First of all, I wish to express my sincere gratitude to ISAMS for the presentation opportunity that they gave me in the conference. It is a great honor for me, a great honor and a pleasure for me to introduce what I have been doing in my laboratory. Especially, there is a lot of interest in gaining a better understanding of how acupuncture works in the brain to influence addictive behaviors. Today, I'd like to talk about the neurobiological mechanism underlying bidirectional acupuncture's effectiveness in the treatment of drug addiction. Drug addiction is a chronically relapsing disorder characterized by a compulsive drug seeking and taking, and the appearance of a negative emotional state, including anxiety and depression during withdrawal from chronic drug administration. Many believe that addiction progresses from two primary sources of drug reinforcement, positive and negative reinforcement. Two major theories of the incentive sensitization theory and the opponent process theory address positive reinforcement and negative reinforcement of addiction, respectively. These two different theories has seemingly opposite views on the role of the mesolimbic dopamine pathway in mediating compulsive drug-seeking behavior. Let me talk about an incentive sensitization theory of addiction as the neural basis of drug craving. Acute drug administration produces incentive salience of drug rewards and drug-associated learning by activation of the mesolimbic dopamine pathway, thereby causing wanting and subsequently repeated drug administration. The mesolimbic pathway is a collection of dopamine neurons that project from the ventral tegmental area in the midbrain to nucleus accumbens in the limbic system. Repeated administration of drugs have the mesolimbic dopamine system enduringly sensitized to drugs or drug-associated stimuli. This causes more wanting, and intense drug seeking is in turn triggered by drugs or drug-associated stimulus. Now I'd like to talk about opponent process for the negative reinforcement of addiction. The negative reinforcement is defined as drug seeking and taking to get rid of a negative emotional state, including anxiety and dysphoria, that accompanies drug withdrawal. This negative emotional state might be responsible for the intense drug craving. The negative emotional state is caused by decrease in reward neurotransmission, such as dopamine in the mesolimbic pathway, and opioid peptide in the nucleus accumbens, and the activation of brain system, including cortical-releasing factor in the amygdala. All addictive, as mentioned earlier, the mesolimbic dopamine pathway projects from the ventral tegmental area to the nucleus accumbens. Moreover, dopamine neurons under individual control of GABA neurons in the VTA. Importantly, all addictive drugs, including cocaine, cause dopamine release in the nucleus accumbens through inhibition of VTA GABA neurons. especially repeated cocaine administration induce neural sensitization in the mesolimbic dopamine pathway, showing that drug evoked dopamine release in the nucleus accumbens is augmented in sensitized animals to abuse the drugs. Importantly, sensitization is intimately implicated in enhanced drug reward-seeking behavior and relapse. This slide shows the inhibition of GABA neurotransmission in the VTA by cocaine. These electrophysiological recordings display that inhibition of VTA GABA neuron by acute cocaine result in enhanced VTA dopamine neuron activity through inhibition of VTA GABA neurons. In contrast to drug sensitization, withdrawal from chronic ethanol administration induces dopamine depletion or reduction in the nucleus accumbens by the hyperexcitability of VTA GABA neurons. The reduction in dopamine release in the nucleus accumbens may represent underlying physical withdrawal syndrome and negative emotional state and ethanol-seeking behavior. From now on, I'd like to talk about our studies we have carried out to date using behavioral and neurochemical paradigm in rats. We evaluated the effect of HT7 acupuncture on drug sensitization. Rats was subjected to manual acupuncture at bilateral HT7 points per 1 minute. The HT7 point on the heart channel has been used to treat mental and psychiatric disorder in clinic. First, let me talk about our previous finding regarding acupuncture and the positive reinforcing effect of drugs. We have previously shown that acupuncture at Sandman HT7 points enhance GABA neuron activity in the VTA, reduce drug-induced dopamine release in the nucleus accumbens, and the behavioral sensitization and the dopamine sensitization to morphine. Additionally, it has been reported HT7 acupuncture reduced the behavioral sensitization to cocaine. Based on these findings, we hypothesized acupuncture at HT7 may reduce relapse to cocaine or stress-induced cocaine-seeking behavior by enhancing GABAergic inhibition of mesolimbic dopamine pathway. To identify effects of acupuncture on stress-induced cocaine-seeking, rats underwent cocaine self-administration in operant chamber, followed by extinction and exposure to intermittent push-up stress. Immediately before push-up, acupuncture was applied at bilateral segment point per 1 minute manually. Results showed that acupuncture at HT7 but not LI5 reduce stress-induced responding on cocaine cocaine powder level without affecting food consumption. This suggests that acupuncture may exert an inhibitory effect on cocaine-seeking behavior by reducing motivational influence, but not motor impairments. Using immunochemistry, we evaluated acupuncture effect on expression of CFAS and the PCREP. which were used as a markers of neuronal activation. Results show that acupuncture inhibited push-up stress induced neuronal activation in the nucleus accumbens. Together with behavioral results, these results suggest that acupuncture attenuated the stress-induced cocaine seeking by decreasing neuronal activity in the nucleus accumbens. On the basis of these results, we tried to investigate whether acupuncture cocaine-induced relapse to cocaine-seeking behavior through activation of GABA neuron in the ventral tegmental area. Results show that acupuncture increased the GABA release in the ventral tegmental area in cocaine-naive rats, and also blocked cocaine-induced inhibition of GABA release in the ventral tegmental area. Results obtained from the microdialysis study demonstrate HT7 acupuncture inhibited extracellular dopamine levels released in the nucleus accumbens in acute cocaine-treated rat, which was blocked by the selective GABA-B receptor antagonist cyclopan. This result implicates that acupuncture modulates dopamine neurons induced by acute cocaine by activation of GABA neuron in the ventral tegmental area. This slide of microdialysis study shows effect of acupuncture on chronic cocaine-induced sensitization of extracellular dopamine levels in the nucleus accumbens. In the nucleus accumbens shell or core, rats were treated chronically with cocaine followed by systemic cocaine challenge after the last daily injection. Results showed the systemic challenge with cocaine hydrochloride produced a much larger increase in dopamine release, which was blocked by acupuncture at H7 but not control point. This suggests acupuncture's role in modulating cocaine sensitization, and this also suggests that acupuncture may reduce cocaine-seeking behavior by inhibition of ventral tegmental area dopamine neurons. We carry out a within-session reinstatement paradigm in rats self-administration cocaine. Each reinstatement test session consists of 4 hours cocaine self-administration, extinction, and cocaine priming. The systemic cocaine challenge induced a significant increase in active level responses in extinguished rat compared to the saline challenge. However, acupuncture attenuated the drug-paired level response without affecting level response for food, which was reversed by cyclopan. This indicates acupuncture may inhibit relapse to cocaine seeking by enhancement of GABAergic inhibition. Second, I'd like to talk about the effects of acupuncture on opponent process for negative reinforcement of addiction. On the acupuncture stimulates beta-endorphin neuron in acute nucleus of hypothalamus. And the endorphinergic fibers can, in turn, activate the opioid receptor on GABA neuron in the nucleus accumbens, resulting in enhanced dopamine neurons activity in the VTA. We have previously observed acupuncture at 87 plays an important role in suppressing the reduction of extracellular dopamine levels in the nucleus accumbens and the behavioral withdrawal sign in alcohol-dependent rat. Based on these points, it was hypothesized acupuncture would reduce ethanol dependence by restoring decreased dopamine release in the nucleus accumbens through activation of endorphinergic neuron input to the nucleus accumbens from the arcuate nucleus of hypothalamus. I'd like to introduce our experimental procedure for induction of alcohol dependence in rat. We used liquid diet and withdrawal. Rats in the ethanol group were exposed liquid diet containing ethanol. Control rats were fed on isocaloric liquid diet without containing ethanol. After 2 hours ethanol withdrawal, behavioral response was measured. In this study, we used mechanical acupuncture instrument. At first, we like to investigate whether acupuncture can activate the beta-endorphin neuron in the arcuate nucleus of hypothalamus. We used the immunohistochemistry method in rat. The results showed acupuncture H7, but not LI5, increased CPAS expression in the arcuate nucleus. Although data not shown, we identified retrograde tracer di-I labeled cell in the arcuate nucleus after we injected di-I into the nucleus accumbens. This suggests neural connection between arcuate nucleus and the nucleus accumbens. In addition, using extracellular recording, we assessed the effects of acupuncture on the putative beta-endorphin neuron activity in alcohol-dependent rat. Extracellular recording shows the basal neuronal firing rate in ethanol diet group are lower than those in the control diet group. However, rats receiving ethanol diet have a greater increase in neuronal firing rate than rats receiving control diet after HT7 acupuncture. Together, results suggest acupuncture can activate the beta-endorphin neuron in the arcuate nucleus. Using microdialysis in freely moving rat, we also evaluated the effects of acupuncture on beta-endorphin release in the nucleus accumbens during ethanol withdrawal. Results showed that acupuncture at H7 increased beta-endorphin release in ethanol withdrawn rat. Similar to beta-endorphin neuron firing rate in the ARC, basal beta-endorphin release in the nucleus accumbens in ethanol diet group was lower than control diet group during ethanol withdrawal. Importantly, acupuncture at H7 increased beta-endorphin release in the nucleus accumbens in ethanol withdrawn rat. Using immunohistochemistry in ethanol withdrawn rat, we attempted to evaluate the effects of beta-endorphin in the nucleus accumbens on dopamine neuron activity in the VTA. After local injection of beta-endorphin into the nucleus accumbens, TH expression phosphorylation as a dopamine neuronal activation marker were measured. Results showed the TH expression and the phosphorylation in the ethanol group were remarkably decreased compared to control diet group, which was blocked by local injection of beta endorphin into the nucleus accumbens. This suggests that intranucleus accumbens of infusion beta endorphin induce the reversal of the hypodopaminergic state during ethanol withdrawal. Interestingly, our other previous studies show that acupuncture at H7 prevented a decrease of dopamine release in the nucleus accumbens in rats treated chronically with ethanol by intraperitoneal injection, followed by ethanol withdrawal. Similar beta endorphin data in alcohol-dependent rat, there was a significant decrease in extracellular dopamine levels in the nucleus accumbens in ethanol group compared to saline group. Using elevated plasmase test in alcohol-dependent rat, acupuncture enhanced time spent in open arm during ethanol withdrawal. Also, this acupuncture effect was mimicked by intranucleus accumbens infusion of beta endorphin. This suggests that acupuncture inhibition of anxiety-like behavior in ethanol withdrawal rats may be mediated by beta endorphin in the nucleus accumbens. Rats underwent alcohol self-administration in operant chamber, followed by 16 days of exposure alcohol diet. After ethanol withdrawal, rats were given the opportunity to self-administer alcohol for measuring alcohol-seeking behavior. Oral alcohol self-administration took place in operant chamber equipped with two response levers. Using oral alcohol self-administration paradigm, we evaluated the effect of acupuncture or intranucleus accumbens infusion of beta endorphin on alcohol-seeking behavior in dependent rat using sucrose fading procedure. Results showed that rats receiving ethanol diet display more ethanol paired level responding than rats receiving control diet. Importantly, acupuncture or infusion of beta endorphin reduce the expected increase in ethanol paired level responding. However, acupuncture or infusion of beta endorphin did not affect water self-administration. In summary, acupuncture may reduce drug craving by correcting both dysfunction of the mesolimbic pathway, hyperactivity in sensitization process, and the hyperactivity in opponent process of drug addiction. Our results open the possibility that acupuncture might activate the adaptive control of central nervous system to restore homeostatic balance. Given the limited time, I cannot discuss the proposal mechanism underlying bidirectional regulation of acupuncture in the mesolimbic dopamine pathway. But our previous data, based on our previous data, our findings provided some insight into the role of the endogenous opioid system. So now we are conducting the possible mechanism of bidirectional role of acupuncture in modulating the mesolimbic dopamine pathway by investigating the role of the endogenous opioid system. Next time, I hope to discuss the possible mechanism underlying acupuncture effectiveness in opioid, endogenous opioid pathway. Thank you for your attention.